Hi everyone. In this lecture, we will cover microbial growth and controlling microbial growth. So the first part of the lecture, I'm going to talk about how bacteria grow in lab and in nature. And then in the second part, we're going to talk about how we control microbial growth. So if, for example, right now with coronavirus going on, everyone is using Clorox wipes and hand sanitizer. And viruses are a type of microbe, so we want to control their growth if they're pathogenic, if they cause disease, such as coronaviruses. And the same thing with bacteria. So we're going to talk about how bacteria grow in lab and in nature, and then what we do to control their growth. So examples such as using various cleaning methods and then pasteurizing milk. We will talk about that. So in the outline of this lecture, we will cover what is needed for organisms to grow, because if we know what is needed for organisms to grow, we know how to control their growth. We're going to talk about terms such as bacteriostatic and bactericidal. I'm going to cover what these terms mean: sterilization, disinfection. What is the exact scientific definition of these terms, and then different physical ways we can control microbial growth, such as UV radiation, and then different chemicals that we use all the time in lab, and then just in our everyday life to control microbes. And then in the end, we will talk about why microbes are becoming resistant to so many different chemicals from the overuse of chemicals to control bacterial growth. So I'm going to start off with bacterial growth in nature. So by nature, I mean naturally. Bacteria can be found either planktonically or in biofilms. Planktonically just means single cells of bacteria, so they're just on their own, independent single cells. And then biofilms means bacterial communities. So biofilms are when you have a lot of bacteria together growing in a community, and I will talk more about this. So these bacterial communities form these slime layers, and you can imagine for infections, these are not a great thing because it's an example of like a layer growing on someone's kidney or someone's lungs, and they help bacteria adhere to surfaces, and these surfaces can be living, such as adhering to each other, or non-living, such as adhering to a table. And also by living, you can also talk about human tissue. So bacterial communities or biofilms, that is the definition of a biofilm. They're very beneficial to bacteria because in these communities or biofilms that bacteria form, bacteria can share nutrients with each other, and these communities also help protect bacteria from different harmful environmental factors. Examples of these are, for example, when you have an infection and the infection is with a biofilm, your immune system creates a lot of white blood cells that go to target that infection, that bacteria. So by bacteria being in a biofilm or in a community, they almost create like a layer to protect each other from all your really great white blood cells that are trying to target the bacteria. And also, for example, antibiotics. So antibiotics are not as effective on biofilms as they are on planktonic cells. And just keep in mind that most human infections are caused by biofilms. So about 70% of human infections, catheters, when people go to hospitals, have surgery and they have catheters in, or people who generally use catheters, one of the biggest risk factors of catheters are infections, so urinary tract infections. And it's because bacteria can form these biofilms or bacterial communities on the catheter that someone uses. So it is a big area of research of creating catheters with various materials that is antibacterial. Heart valves, contact lenses, cavities, all of these things are also caused by biofilms. And then in digestive system and sewage treatment systems, we can see biofilms clogging these systems. So digestive system not working as effectively, or sewage systems having so much clogging because of these bacterial communities. And here we see in this image that bacteria can start growing planktonically, and then they form a biofilm. So they all work together to basically form this community that is really helpful for the bacteria, but not great for us because they're very hard to treat. And they are generally resistant to a lot of different drugs, a lot of different antibiotics. So we really this is why it's important when someone has a bacterial infection, if possible, to treat it early on before the bacteria overgrow and we have this biofilm or bacterial community. And we will learn later on about cavities or dental caries, and this is caused by a biofilm from bacteria forming on teeth, and these bacteria produce so much acid that erode the teeth. Okay, let me go on to the next slide. So bacteria communicate with each other in these communities by a process called quorum sensing. I want everyone to know the definition of quorum sensing. Quorum sensing is bacteria cell-to-cell communication, and they communicate with each other by secreting signaling molecules that we call autoinducers. These signaling molecules are autoinducers. So signaling molecule and autoinducer mean the same thing. What they do is they cause the cell to turn on or off a particular gene. That's what we mean by expression of particular genes. So let me explain it more. So when you have bacteria, a lot of bacteria next to each other, a bacterial community or a biofilm, bacteria will produce these signaling molecules called autoinducers. So you have a bacteria, it will produce an autoinducer, and the autoinducer can be recognized by other bacteria. So this signaling molecule will be recognized by other bacteria. The signaling molecules a lot of times will bind on a receptor on the bacterial cell and then tell the cell to do something, such as create a pigment to protect yourself, or move, or create a biofilm, stick to each other. So this is what quorum sensing is. It's a big area of research of bacteria communicating with each other. And the reason why this is important in terms of the medical field is that if we can stop bacteria from communicating with each other and forming biofilms, we can potentially treat a lot of different infections that can be much more virulent or worse because of the biofilm. So again, with the way that quorum sensing works is that bacteria communicate with each other by producing these signaling molecules, which we call autoinducers. And then what autoinducers do is they induce the expression of various genes, such as biofilm production or pigment production. So in one type of organism, we see that when autoinducers are produced, they signal the bacteria to form bioluminescence. So we see that. So quorum sensing is a big area of research, and here we see quorum sensing in lab. So here is Pseudomonas bacteria, Pseudomonas aeruginosa. And when we added Pseudomonas aeruginosa, which does quorum sensing, and another bacterial extract, we see that all the autoinducer or signaling molecule induces the bacteria to produce a biofilm. So you see this thick layer of bacteria on this slide in lab. Okay, now we're going to talk about how we grow bacteria in lab, and I want everyone to know a few terms. So the term sterile means that there's absolutely no microbes. So when we say no one is sterilizing anything at home, we are cleaning at home. A lot of times people misuse this word. We sterilize things in lab and we sterilize equipment in hospitals. Sterilization takes a lot of different methods to make sure that there's absolutely no living microbes. So the word sterile is a very strong word versus clean. Just know that sterile means there's absolutely no living microbes. And that's very hard to accomplish, to make sure that you've absolutely killed everything. The word inoculate means to put microbes somewhere. So if I tell you inoculate this tube or this flask or this plate, it just means put bacteria or put microbes onto that medium. The word culture means microbes growing in or on a medium. So if I tell you this bacterial culture, it's just the bacteria itself. Culture medium, the word medium means what we grow the bacteria on. So if you're growing, media is plural, medium is singular. So if you're growing bacteria in lab and I ask you what medium did you use, you could tell me I used blood agar, for example, or I used broth. So medium is just a fancy word for saying what you exactly grew your bacteria on. Different types of media we use in lab are broth and agar to grow bacteria in. So broth, just like what you think of with chicken broth or vegetable broth, is the liquid form of a media. And this is important when we grow bacteria, because in the liquid form, we can see turbidity or cloudiness of a culture, and this indicates that bacteria grew. So if you have two tubes and you're not sure if bacteria grew and you see cloudiness or turbidity, it means that bacteria grew. This is also another way to think about this. So this is in lab, is Urine should be transparent. If you see a patient's urine and it's cloudy or turbid, it means that bacteria grew. So they probably have an infection. And then another form of media that we use in lab is agar. Agar is just a solid media. So it looks like a jello. That's probably the best consistency I can think to explain it to you since you're not in lab looking at it. So it's like a layer of jello that we typically have agar growing on plates. Sometimes we use tubes and we do these slants, but typically it's in these plates. So that's what we call an agar plates. And the media is this nutrient-rich jello. That's the best way I can explain it. And we inoculate bacteria on agar, and when we inoculate bacteria on agar, we can see colonies. So we can see colonies that indicate bacterial growth. So for the differences in media, in liquid culture, in broth culture, we know that bacteria grew if we see turbidity or cloudiness. And then on agar, we know that bacteria grew if we see colonies of bacteria. So this is what a plate would look like. And both are used in lab depending on what you want to do. So if I'm just looking to see if bacteria grew, maybe a broth culture is fine. And it also depends on the experiments that you're planning to do. Okay, now we're going to talk more a little bit about agar. So agar, again, it's a solid medium, so it's not like broth, it's not liquid, and it looks like jello. It's a complex carbohydrate, it's a complex polysaccharide that has extracts of yeast, meat, or plants. It basically is a very nutrient-rich medium for bacteria to grow on, and we put them in Petri plates. The little round plates are called Petri plates and slants. It's generally not metabolized by microbes, so microbes grow on it, but they don't degrade it. And it's liquid when it's really hot. That's how we make it in lab. And then when it gets cool, it solidifies, exactly like jello, if you've ever made jello at home. And we use it to look for colonies of bacteria. So if we look at this agar plate, I can see that this agar plate is not a pure culture. It's a mixed culture. Pure culture means we only see one type of bacteria, but I see these kind of yellow colonies and then these more like whitish colonies. So I know that this is not a pure culture, it's a mixed culture. So I want everyone to know the definition of pure culture. Pure culture means that there's only one species of bacteria. So if someone has an infection and maybe they have a throat infection and you suspect that it's a bacterial infection, and you take a sample and then you inoculate an agar plate, meaning you swipe it on an agar plate, and you come the next day and all the colonies or these little circles are all, let's say, green colonies, that I know that culture is pure. But if I see different colors, that I know that the patient has an infection with different types of bacteria. And then we talked about colony a little bit in the lab lecture. A colony just means that you have one species of bacteria that gave rise to millions of bacteria. So you see that little circle. So this we call a colony, that's a colony, that's a colony. So it's a population of cells that arose from a single species of bacteria. So in this colony, there's millions of bacteria of the same species. That's the key word, same species. And a lot of times students tell me, is that one bacterium? And the answer is no. Remember, we cannot see a bacteria with our naked eye, but I can see this with my naked eye. So for me to see this, it means that there are millions of bacteria in that really, really tiny little circular colony. It's also called a colony forming unit, and keep in mind, it's millions of bacteria that originated from the same species. So in lab, we use something called the streak plate method, and I have a lab video explaining that. We use that method to isolate colonies of bacteria. So this is the streak plate method. And then you see here, you basically dilute bacteria until you can see individual colonies on a plate. So the take-home message from this slide is, when we grow bacteria on agar plates, we're looking for colonies. So if a scientist is specifically interested in seeing if a culture is a pure culture or a mixed culture, you can just streak it on an agar plate, and if all the colonies don't look the same, you know it's a mixed culture. But if they all look the same in terms of like texture, shape, color is a big indication, then you know it's a pure culture. Now, for growing bacteria in broth culture or liquid culture, the broth culture is similar to agar. It's very nutrient rich. So you want to make sure that whatever you grow your bacteria on, whether it's agar or broth, that medium has all the nutrients that we need for growth. And remember, the 4 macromolecules that are needed for growth are carbohydrates, proteins, nucleic acids, and lipids. So it's a mixture of all of those things. And to know that if bacteria grew, we look for turbidity. Turbidity means cloudiness, meaning that something is not transparent. So this is typically, like if you look at this tube too, right here, broth without any bacteria is very transparent. And then when you put bacteria in it, when you inoculate bacteria and then you go incubate it. Incubate it means you put it in this little oven that's warm so bacteria can grow faster. And you come back the next day in lab and you see this cloudiness or turbidity, you know that your bacteria grew. And so when we see cloudy urine, it indicates that someone has an infection. So this is very important. That's why when you're taking care of patients or you yourself see that your urine is cloudy or smelly. Smelly means there's bacteria that's doing metabolism and producing gases. It means there is an infection because urine should not be cloudy. So now we're going to talk about bacterial growth, and I just want to go over a few terms. So bacterial growth means an increase in the number of bacteria, not cell size. When we think like human growth, we think that the human got bigger. For bacteria, the word bacterial growth means an increase in cell number. So bacteria grew if you went from 1 cell to 2 cells or 1 cell to 500 cells. That's what bacteria growth. It's an increase in bacteria, in the number of bacteria, not the cell size. So this is a test question I like to ask. And bacteria grow by a process called binary fission. Binary fission means the bacteria just divide. There is no sexual reproduction. So that's why bacterial infections can evolve so rapidly, because they can easily divide or do binary fission. So here's binary fission. So bacterial cells, if it's going to divide, it just makes a copy of its genetic material, its DNA, and then it just divides. That's binary fission. And bacteria grow exponentially. Exponentially means that 1 bacterial cell becomes 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32. So you guys get the idea. And the time it takes for a cell to divide, a bacterial cell to divide or double, in science we call that the generation time or doubling time. It means the same thing. So doubling time seems like a better term for it, but we say generation time a lot of times. It just means that how long it takes for a bacterial cell to divide or double. So for example, E. coli is a type of bacteria, and for 1 bacterial cell, for 1 E. coli to become 2, it takes about 20 minutes. For 2 to become 4, because bacteria grow exponentially, it takes another 20 minutes. For 4 to become 8, it takes another 20 minutes. So if a scientist says, what's the generation time or doubling time of E. coli, it's 20 minutes. Every bacteria has a different generation time. For example, the bacteria I used to work with, H. pylori, it's 5 hours. So it's a lot slower to grow than E. coli. And then I always like to tell students about Mycobacterium tuberculosis. So Mycobacterium tuberculosis takes 20 hours for 1 bacteria to become 2, 2 to become 4, 4 to become 8. It's very slow growing. So for people who do research on Mycobacterium tuberculosis, research is very slow on this disease and this bacterial organism because it just takes this bacteria so long to grow. So also with infections, with people having tuberculosis infections, they take antibiotics for a very long time. And we'll learn all about that later. But I want everyone from this slide to understand that binary fission is when bacteria divide, and the time it takes for bacteria cells to divide or double is called generation time, and every bacteria has a different generation time or doubling time. So when we grow bacteria in lab, whether you're growing it in broth culture or agar culture, you can form something called a growth curve. And a growth curve shows the growth of bacteria over time. So if we were in class, I would draw this on the board. But basically, you have a graph. So you have your y-axis and your x-axis. And your x-axis is time. So just over time what happens to bacteria. Your y-axis is the log number of bacterial cells. So it's a number of bacterial cells, and bacteria grow exponentially, so that's why it's on a log scale. And so there's 4 phases of bacterial growth. When we draw a growth curve, and I'll explain this in a second, but it's very important that you know the 4 phases of bacterial growth. When we first inoculate a medium or put bacteria in a medium, whether that's your agar plate or your broth, this is what happens to bacteria. First, in the beginning of time, so in the beginning you have your lag phase where bacterial growth is lagging because there's high metabolism and bacteria are still getting accustomed to the nutrient you put it in. Then once they get accustomed, we see the log phase of bacterial growth. So log or exponential phase, people use both terms, is when bacteria are doubling, and then bacteria will divide, and then we see a stationary phase where they're dying and also being created at the same time. So it's an equal rate. We call that stationary phase. And then eventually over time, bacteria will start dying because of all the toxic components that are being made. And then there's not enough nutrients in the broth or the agar that you grew the bacteria in. So this is a growth curve. I want everyone to know the 4 phases and how to draw this if you're asked to draw this. If we were in class, it would be much easier because I would draw this for you. But again, the key to drawing this is your x-axis is time. That's how you would label it. Your y-axis is the log number of bacteria. And then the growth curve, the first phase of bacteria is the lag phase where there's little to no cell division or binary fission because bacteria are still getting accustomed to the media that you put it in. So we see high metabolism. The next phase in our growth curve is the log phase of bacteria. So this is where bacteria are exponentially dividing. So we see high cell division, and then stationary phase where we see that the rate of bacterial growth equals the rate of bacterial death. And then finally, the death phase where we see bacteria start to die because there's not enough nutrients and there are a lot of toxin byproducts being formed as byproducts of metabolism. So these are the 4 phases of bacterial growth: lag phase, log phase, stationary phase, and death phase. And I want you to know what happens in each phase. So in the beginning, in lag phase, we see high metabolism, little to no cell division. In log phase, we see high cell division. In stationary phase, the cell division rate is the same as the death rate. And then in the death phase, bacteria are dying because of lack of nutrients and toxin production. So with the growth curve, we're seeing a growth curve in a closed system. A closed system meaning in lab, you're looking at bacteria on broth or agar. Typically we do this with broth, but you can avoid death phase if you supply the agar with nutrients again. So that's what a continuous system is. So I want everyone just to keep in mind what I said in the last phase about the different phases and what happens at each phase. What happens at lag phase, log phase, stationary phase, and death phase. So now that we know a little bit about the growth curve, we're going to talk about the requirements of growth. What do you need for microbes to grow? And microbes, just like humans, need important things. So they need food, which is nutrients, and they need the right environment. So the temperature has to be right. All different, I'm going to talk about bacteria specifically, but we can think of viruses as well, since coronaviruses are a big topic right now. So you need the right temperature for every microbe to grow. I'm sure some of you have heard in the news that coronaviruses prefer cold temperature over warm temperature. That applies to different bacterial species. Every microbe has a different temperature it likes, but I will say that most bacteria, specifically for bacteria, not viruses, like warm temperatures. Also, different microbes like different pHs, but most microbes like neutral pH, not acidic, not basic. And then osmotic pressure, we want to make sure that we have the right salt and sugar concentration for our microbes to grow. And then oxygen. Some microbes are aerobic and they need oxygen. Some microbes are anaerobic and they're killed by oxygen. Some microbes can grow aerobically or anaerobically. So we'll talk about that. And then nutrients are very important. Remember that we said that when we look at a cell, whether it's a all cells, whether it's a bacterial cell or a human cell, we have 4 macromolecules that make up cells, which are carbohydrates, proteins, lipids, and nucleic acids. And so to make these things, you need the different elements. So we want to make sure that we supply carbon, nitrogen, sulfur, phosphorus, trace elements, oxygen. So make sure you have food for your microbes to grow. If you take away food, microbes won't grow. So we're going to start off by talking about temperature. So all bacteria have an optimum temperature. Optimum temperature means the best temperature that they have the highest growth at. Phile means love. Some organisms are psychrophiles, means that they love cold temperatures. So their optimal temperature is 10 degrees Celsius. And these are the microbes that cause our food to spoil in the fridge. So the microbes that can grow in the fridge like cold temperatures, so they're psychrophiles, they're cold loving. Mesophiles, which are most microbes, are moderate temperature loving bacteria. Their optimal temperature to grow in is 37 degrees Celsius, which is body temperature. Most microbes like moderate temperature. Thermophiles are microbes that like heat, so their optimal temperature is 60 degrees Celsius. So these are microbes that we see growing in very warm places. And then hyperthermophiles, which are rare to find, and microbes are microbes that like really, really high heat. So their optimal temperature to grow at is 95 degrees Celsius. So in the deep ocean, we see hydrothermal vents. And in hydrothermal vents, this is extremely hot. We see some microbes that grow there, and so the microbes that grow there are hyperthermophiles. Just know every bacteria has an optimum temperature, and if I tell you a bacteria is a psychrophile, I want you to know that it's a cold loving bacteria. If I tell you it's a mesophile, it means that it likes moderate temperatures, 37 degrees Celsius. So go through these, but just know that in general, most bacteria are mesophiles, most like to grow at body temperature. And so the bacteria that are hyperthermophiles, I'm just going to talk about this. You may ask yourself, because we learned about it in a previous lecture, that protein is very important for cells to function, and proteins are denatured at high temperatures. That's why fevers are bad. So how do bacteria that are hyperthermophiles survive in hot environments? And the answer is the bacteria that are hyperthermophiles, or thermophiles in general, have special proteins that are folded in a way that does not easily denature at high temperatures. So their folding is a little bit more, just a little bit more complex, so that they can survive in these hot temperatures. So I want to go over these terms, bactericidal and bacteriostatic. Bactericidal means any temperature that kills bacteria, and bacteriostatic is, if we're talking about temperature, a bacteriostatic temperature is a temperature that stops bacterial growth. So high temperatures are typically bactericidal. They kill bacteria. That's why we cook food. Low temperatures, refrigerator, is bacteriostatic. It stops bacteria from growing. It does not kill bacteria. That's why bacteria in the refrigerator eventually goes bad. It slows down the growth. So this is an example of why bacteria will eventually go bad, whether it's left at room temperature or you put it in the refrigerator, but it will last longer in the refrigerator because the refrigerator is bacteriostatic. It'll just stop the bacteria from further growing. And we'll talk more about this in a little bit, about these terms. Another important thing to consider when we're thinking how to grow bacteria or microbes in lab is pH. Most organisms like neutral pH, so pH of 7 or pH between 6.5 to 7.5. Just know most organisms just like a pH of 7, and so these are neutrophiles. There are some rare circumstances where microbes are acidophiles and they like acidic environments, and then some microbes that are alkalophiles that like basic environments. So this is a reason why also we pickle foods. Pickle foods do not spoil fast, they last forever, and it's because you put food in this very acidic environment, and most microbes do not like acidic environments because most microbes are neutrophiles, so it's a way of controlling their growth. And this is also the reason why we've evolved where our stomach pH is so low. So your stomach is very acidic for 2 reasons. 1, so that it can digest food, and 2, so that it doesn't grow microbes, because most most microbes cannot survive in the stomach. Most microbes need a neutral pH, and the stomach pH, the acidity of the stomach, kills bacteria. So in lab, we use something called buffers to control pH. So when you're growing bacteria in various media, whether that's agar or broth, we add buffers, which are just chemicals that try to keep the pH around 7. And the question would be, well, how do bacteria that are acidophiles or alkalophiles? How do they survive in these environments? And the answer is that the bacteria that are acidophiles or alkalophiles generally have enzymes that allow them to survive in very acidic environments or very alkaline environments. So an example, H. pylori. H. pylori is a bacteria that causes stomach infections, different types of stomach infections. And the reason why it can survive in the stomach that is very acidic is because bacteria produces an enzyme called urease, and urease neutralizes the environment around it so that it brings the pH back a little bit higher, so it's not as acidic, because the low pH, the acidity, would generally kill bacteria. Another thing we consider when growing bacteria in lab is osmotic pressure. So osmotic pressure has to do with the solutes we put in lab to grow bacteria. Remember that bacteria can be grown in 3 types of solutions. We have hypertonic solutions, isotonic solutions, and hypotonic solutions. The type of solution that bacteria generally like to grow at is an isotonic solution, because hypertonic solutions have such high salt concentrations that they will cause cells to lose all their water and shrink and die. And so that is not a great type of environment to grow bacteria in. This is why we use salt as a way to preserve food, salts and sugars. So jams, so fresh strawberries outside will eventually go bad, but if you have strawberries in jam form, jam has a ton of sugar in it, they can last for years and nothing will happen to jam. The reason is that there's so much sugar in jam that bacteria cannot grow in it. In general, they cannot grow because it's a hypertonic environment. This is the same idea where we see salted anchovies. So if you just have anchovies outside, seafood goes bad very quickly. But when we do high salt concentrations to preserve food, we don't see bacterial growth because bacteria do not like that hypertonic environment. And it's the same thing with hypotonic environments. Hypotonic environments are also not a great thing. Now, most organisms do not like high salt, but there are exceptions, just like we saw with pH and temperature. So there are organisms called halophiles. Halo means salt, phile means loving. Obligate halophiles are organisms that require very high osmotic pressure, high salt to grow. So they cannot grow unless you have high salt. And a lot of, for example, ocean microbes are obligate halophiles. There are also facultative halophiles. These are organisms that can tolerate different salt concentrations. They're not killed by it. An example of facultative halophile is Staphylococcus epidermis. This is the bacteria that we see growing on our skin. So your skin has so many different salt concentrations, for example, when you sweat and throughout the day. And so Staphylococcus epidermis is able to tolerate different salt concentrations. It's not killed by it. So again, just to recap, sugars and salts are used as preservatives because most organisms do not like hypertonic environments, but there are organisms such as halophiles, and the way that they prevent lysis in high salt environments, because high salt environments are hypertonic that can cause plasmolysis, is that they usually have pumps in their cell membrane that will pump the salts out. So that's one way of dealing with high salt concentrations. Another environmental factor to consider when we grow bacteria in lab is oxygen. So different organisms, some like oxygen, some are killed by oxygen, some can grow everywhere. Organisms that are obligate aerobes require oxygen for respiration. So as an example, humans, we're obligate aerobes. We need oxygen to survive. So obligate aerobes are obligated to have oxygen. Obligated means you have to. Most bacteria are obligate aerobes, and just keep in mind that oxygen has a lot of toxic byproducts. So in order to be an obligate aerobe, those organisms usually have the enzymes catalase and superoxide dismutase, which is sometimes just abbreviated SOD. Organisms that are obligate anaerobes, anaerobes means no oxygen, are killed by oxygen, and they can only grow when there's no oxygen present. So research labs that work with obligate anaerobic organisms struggle a lot to work with them because they're not easy. Any tiny bit of oxygen that gets in will kill the microbes. So you have to make sure to work in a hood that's sucking up all the oxygen. Facultative anaerobes are organisms that can grow in oxygen and without oxygen. So they can do all types of metabolism. They will grow, they will do aerobic respiration when oxygen is present, but then if there's no oxygen, they're able to do fermentation or anaerobic respiration. And then aerotolerant anaerobes are organisms that can tolerate oxygen, but they do not use it for their respiration. So if you were growing these organisms in lab, if you were growing them in special tubes, so thioglycolate tubes, and I'll talk about it in a different lab video, obligate aerobes will grow at the top because they require oxygen to grow. Obligate anaerobes will grow at the bottom because they are killed by oxygen. Facultative anaerobes will grow everywhere, but we'll see the most growth at the top because they would prefer aerobic respiration over anything because you produce the most ATP. Aerotolerant anaerobes tolerate air but are anaerobic. And then microaerophils, I'm going to talk about this. These are organisms that require oxygen in very low concentration. So H. pylori is a microaerophil, for example. So they grow just a little bit under the highest level of oxygen. And if asked on a test, how do obligate aerobes deal with the toxic byproducts of oxygen? It's because they have enzymes such as catalase and superoxide dismutase. So again, here are our different microbes. I want everyone to know the difference between all of them, but the take-home message is obligate aerobes require oxygen to grow. Facultative anaerobes can grow anywhere. They can grow aerobically or anaerobically. And then obligate anaerobes are killed by oxygen. So they can only do fermentation or anaerobic respiration. So if I were to summarize the different types of respiration that these do, obligate aerobes will do aerobic cell respiration. Facultative anaerobes will do aerobic respiration, anaerobic respiration, and fermentation. They can do all three, but if given the choice, they would do aerobic respiration first, then anaerobic respiration, then fermentation. And then obligate anaerobes can only do anaerobic respiration or fermentation. And again, in lab, they're very hard to work with obligate anaerobes because any tiny bit of oxygen will kill them. So we typically, if you are in a research lab that works with an obligate anaerobic bacteria that causes infection, you can work with it using an anaerobic chamber that sucks up all oxygen, or you can use these anaerobic jars that have chemicals inside that suck up all oxygen. Okay, and then finally, when we grow bacteria in lab, we want to make sure that nutrients are present so that they can grow effectively in lab. So their bacteria have major elements that they require, and they have trace elements. Major elements that we see that are important are carbon, nitrogen, phosphorus, and sulfur. And just to remind everyone, carbon is part of all macromolecules. That's why we call macromolecules organic molecules or organic biological molecules. The word organic means that they're carbon-based. So we need carbon to make proteins, carbohydrates, lipids, and nucleic acids. It's an important energy source. And so carbon is a very important major element. Now, two terms I don't want to really focus on this, but I want everyone to know the difference between a heterotroph and an autotroph. A heterotroph uses organic carbon sources. You have to provide it for it. You have to give it food. An autotroph can make its own food, so it uses carbon dioxide, which is an inorganic carbon source. Another major element that all organisms need to grow, so what you have to make sure your agar plate or your broth has in it, is nitrogen. The reason why nitrogen is important is you need nitrogen to make amino acids, which make protein, and nitrogen is an important component of the nitrogenous bases. So adenine, guanine, cytosine, thymine, uracil that we see in DNA and RNA. And also phosphorus is important because we see phosphorus in DNA, RNA, and ATP, and membranes. So cell membranes are all made up of phospholipids, so we need phosphorus. And then sulfur is also an important element because it's part of some amino acids, so it's part of cysteine, for example, and this is important to make various proteins. Also, bacteria, besides the major elements I talked about, they also require trace elements. Trace elements are elements that you need in small amounts. So they're 100% required, but you only need them in small amounts. So this applies to the microbes just like it applies to humans. So iron, zinc, potassium, magnesium, calcium, and they're important for various reasons. But one reason we need these trace elements or these elements in very small amounts for bacteria to grow or humans to grow is because a lot of times they function to help enzymes. So we call them enzyme cofactors. And remember that all cells would not function without enzymes. So they're very important. Okay, so we spent the last few slides talking about how we grow bacteria in lab and what's important for growing bacteria in lab. So now that you know the important things, so such as temperature, pH, oxygen requirements, solute concentrations, once you know these things, you know how to control microbial growth or stop microbes from growing. So that's what we're going to focus on. So the word controlling microbial growth can mean two things. It can mean killing microbes, and we call those cidal agents. So bactericidal agents are things that kill bacteria, or you can control microbial growth by inhibiting their growth, and we call those static agents. So bacteriostatic means something that stops bacteria from growing. Bactericidal means something that kills bacteria. So I want everyone to know these terms. This will definitely be a test question. I will ask you to define bactericidal and bacteriostatic. And again, if we say something is bactericidal, we mean that it kills bacteria. If we say something is bacteriostatic, it stops bacteria from growing. So for example, Clorox wipes are bactericidal for many bacteria, and bacteriostatic is something like refrigerating food. You stop bacteria from growing. So we can control microbes from growing. And I keep talking about bacteria, but know that microbes can mean viruses, can mean bacteria, mold, protozoa, many different microbes. We can control them by either physical methods or chemical methods. Physical methods are like using sunshine or filters. Chemical methods are using chemicals to control their growth. These are terms also I'm going to stop by to explain. I want everyone to know the difference between sterilization, disinfection, and antisepsis. So the word sterilization, it's being used a lot right now in the days of coronaviruses, and I think it's being misused a lot. To sterilize something means to remove and destroy all microbial life. So something is sterile, there are 0 microbes on it, there's nothing on it. And I always ask students in class, can something be a little sterile or a lot sterile? And the answer is no. You're either sterile or not. So sterile means that you have 0 microbes on you. So you can imagine that when you're using disinfectant wipes, you are not sterilizing the surface. You are disinfecting. Sterilizing means that there's absolutely no microbes growing there. To sterilize a surface or like equipment for surgery, you have to go through a lot of different methods so that you can remove every single microbe, including endospores. So the term that is a better term is disinfection. Disinfection means when you destroy harmful microbes on objects or surfaces. It does not mean you remove every single microbe, but you remove most. So you remove harmful surfaces from objects or surfaces. When we remove harmful microbes from living tissue, we call that antiseptics or antisepsis. So mouthwash is antiseptic because you're removing microbes from living tissue from your mouth. Clorox wipes or any other cleaning wipes are disinfectants because you're working with a surface. And then sanitization just means reducing microbes to safer levels. It does not mean sterilization. Again, sterilization is a big word to use. So a few more terms. Asepsis means being free of microbial contamination. So in lab, we use aseptic technique, and aseptic technique is also used in hospitals, and it's used to prevent contamination. We make sure that in hospitals, when healthcare personnel are doing surgeries or doing anything with a patient, they're covered up, they're wearing gloves, they're make sure that they're cleaning masks, just to avoid any contamination. And in lab, we talked about that with sterile techniques. So with aseptic technique, you're just making sure that the loop that you're using to transfer bacteria, you're passing it through the flame, you're wiping your surface. So aseptic technique is very important to prevent contamination of your surface or of the patient. So I like to talk about this in class. So we all use so many different things to control microbial growth. There are millions of things, but these are a few I could think of. So the reason that the people who use contact lenses use contact lens solution is so that you don't have microbes growing on contacts. This is the reason why you don't just put contacts in water. You can put contacts in water. I'm sure people have done it for a few days, but you really want to make sure to use a good solution that prevents microbial growth. Another thing that people use is deodorant. So deodorant has different chemicals in it to prevent bacteria or different microbes from growing. And it's the bacterial growth that causes the smell. So bacteria, people who tend to notice that they have a smell under their arms, it's because bacteria are growing there. And it is just as effective to use deodorant as to use alcohol. So if you just take a little bit of rubbing alcohol and you put it under your arms, it's as effective as deodorant. Acne products, acne is typically caused by bacteria. So we use different acne products to control their growth. And then disinfectant wipes. So disinfectant wipes are a very important one, especially these days, because there's so many chemicals inside to stop bacteria and viruses from growing. Hand sanitizer has a ton of alcohol in it, most hand sanitizers. Catheters can cause microbes to grow, as we talked about. So there's a lot of research being done on the best plastic to use to make sure that bacteria don't grow on catheters. And then antibacterial hand soap that people use. So this is from January this year, the FDA came up with a statement telling Purell hand sanitizer to stop making these crazy claims that 99.9% of microbes are stopped by hand sanitizer. So I just want to talk to you about that. All of these things are in a constant area of research. So Johnson & Johnson works on making a lot of these things, whether it's contact lens solution or deodorant or acne products or hand sanitizer. And what microbiologists are doing in these companies is figuring out new ways to add chemicals to stop microbes from growing. So whenever you're creating a treatment, whether you're creating an acne medication or a new deodorant or catheters or Clorox wipes. So the Clorox company, I don't know if many of you guys know this, but the Clorox company is here in the Bay Area. And so they have a very high area of research of scientists working on this treatment. When you're creating a treatment, again, let's compare Clorox wipes to the next best acne medication. You want to think of a few things. You want to think of the type of microbe you're treating. So companies that are working on acne medication are working on how do we target acne bacteria. Maybe companies right now that are working on the best new Clorox wipe are thinking of coronaviruses. So viruses and bacteria are different things. So you have to consider what is the type of organism I'm trying to target. Or like underarm bacteria is different than the bacteria that maybe causes different types of digestive illnesses. We also want to consider the number of microbes present. So when you're trying to target acne bacteria, that's going to be different than the number of microbes present on a surface where you're using a Clorox wipe. Treatment, also how effective it is, depends on the environment. So it depends on whether you're using a Clorox wipe in an elevator or at home on a kitchen counter. So this is important where the microbe is growing. The effectiveness of treatment is the length of exposure. So maybe someone uses acne treatment for a day and maybe someone uses it for a year and they see different results. Same thing with Clorox wipes. A lot of people don't know this, but with Lysol and Clorox wipes, for them to be effective, let's talk about Lysol. You're really supposed to spray it on your surface and keep it there for a few minutes before wiping it. A lot of people will wipe it right away and it's not as effective if you wipe it right away. And then risk of infection, you want to make sure that what you're creating is not toxic. So if you have people that are immunocompromised, if you have, for example, pregnant women, they use your new acne medication? I mean, a lot of acne medications should not be used by pregnant women, for example, or your new Clorox wipe. And then we want to think of the composition of the item that we're using it on. So are we using it on plastic? So is your Clorox wipe being used on surfaces or on lungs or on people's faces or on people's underarms? And then you also want to consider how toxic what you're making is. So there are a lot of really great acne medications that are also super toxic to humans. So for example, some of you may have heard of benzoyl peroxide and salicylic acid. So if you use various concentrations of these for a very long time, for example, pregnant women and immunocompromised people may not be able to use everything because it could be toxic to them. And so whenever you're creating a treatment to control microbial growth, you want to think about all of these things before you see how effective your treatment would be. You're not going to treat, if you're a company working on strep throat and creating a treatment for strep throat, you're not going to treat it like coronavirus. And it's the same thing, you're not going to treat it like bacteria that cause eye infections if you're creating the newest, best contact lens solution. And then an important thing is cost and availability. I always like to tell my students that your treatment is not that good if it costs $5 million and only a few people in the world can access it. So you want to make sure that what you're creating is accessible to the general public. Okay, so now we're going to talk about the actions of various microbial control methods. So why does heat kill bacteria? Why do chemicals affect microbial growth? Why does radiation? So we're going to learn about what these antimicrobial agents specifically do. So a lot of times antimicrobial agents, whether they're physical such as heat, chemical, or also physical such as radiation, they'll alter membrane permeability. So if you change the permeability of bacteria or viruses, bacteria and viruses may not be able to grow or may die. So that's a good thing if you're trying to target something that is potentially pathogenic. And then a lot of antimicrobial agents damage proteins. Heat damages proteins. That's why we use it to control microbial growth. This is why no one eats raw chicken, or you shouldn't be eating raw chicken. We cook it to make sure that we damage all the proteins of, for example, salmonella that could be contaminating chicken. You could also damage DNA and RNA, which are nucleic acids. So we can achieve all of these things by either using chemical or physical methods. And again, it depends on what you're trying to treat. So examples of physical methods that we use to control microbial growth are heat, filtration, and radiation. And I'll talk about all of these. Examples of chemical methods are like Clorox, alcohol, bleach, all of these things. Now, I always ask students that if you create a microbial control agent and it affects plasma membranes, would it affect humans? And I want you to think, do humans have a plasma membrane? And the answer is yes. So you always want to make sure that when you're doing something, it's harming the microbe and not the human. So it's an important consideration. Okay, we're going to talk about heat. Heat has been used for ages by different societies to control microbial growth. The reason why heat stops microbes from growing is because it denatures proteins. Enzymes are proteins, and enzymes are important. If you stop proteins from folding properly, that's what denaturing means, they will not function properly. So heat destroys or denatures proteins and also destroys other cell components. And if available, it is number one method of choice for microbial growth. So examples of heat are flaming and ovens and boiling. What temperature do hospitals tend to be? I always ask students, and students tell me that the hospitals are typically cold. So we're talking about heat now, but I want everyone to know that hospitals are generally cold because bacteria do not like cold environments. And then why can you eat raw fish but not raw chicken? This is an important question to consider. Now, when I say raw fish, I mean fresh raw fish, not raw fish that's been sitting out for 5 hours. The reason why we can eat raw fish and not raw chicken is because raw fish comes from a very salty environment. And so that will help control microbes for a little bit, but you have to eat it very soon. But you cannot eat raw chicken because raw chicken is not in a salty environment. So microbes like raw chicken, it's very nutrient rich. That's why we have to cook it, and we have to cook it so we make sure that we denature the proteins and destroy cell components. This is also, this slide is not showing, but I wanted to talk to you again to refer back to a previous slide that high temperatures, cooking is typically bactericidal. It's used to kill bacteria. Low temperatures are bacteriostatic. They stop bacterial growth. So this is the idea of milk at room temperature versus refrigerator. Milk will grow bacteria over time, whether it's left out or in the refrigerator. It's just a refrigerator will slow it down. So the refrigerator is bacteriostatic. It stops bacteria from further growing. Another way that we control microbes is by doing pasteurization. Pasteurization means flash heating. Flash means very quick heating. We do about 15 seconds. So we flash heat milk or juice. So juice is also pasteurized, as are other things, but usually we think of milk. And the reason why we do this is to reduce organisms that can spoil food and also pathogens. So that's what pasteurization does. Basically, milk goes in and it goes through a quick heating section for a few seconds and then a cooling section, and then it's pasteurized. So ultra pasteurized milk, if you ever go to the supermarket, an example of ultra pasteurized milk is Horizon milk. It says ultra pasteurized means it's highly pasteurized. If you look at ultra pasteurized milk versus raw milk is not pasteurized. Raw milk has an expiration date that's much faster than ultra pasteurized milk. Ultra pasteurized milk can last for like a month or 2 months in the refrigerator because you've really highly reduced microbes that can cause food to go bad, and as well as pathogens, which are microbes that cause disease. Another physical way that we control microbial growth is through the autoclave. So I want everyone to know what an autoclave is. An autoclave typically looks like this. It's this machine that we see in research labs and hospitals. And there are these machines that go up to very, very high temperatures with very, very high pressure. And this high temperature, high steam, high pressure will sterilize. Remember the word sterilization. They will kill all microbes and endospores. So they're used in hospitals and labs, and they kill about 99.9% of organisms and endospores. So this is where if you're doing surgery equipment is sterilized by using an autoclave. And typically, an autoclave runs at 121 degrees Celsius at 15 psi for 15 minutes. So if any of you guys know anything about psi, it's like your car tire pressure. So 15 psi is pretty high pressure, and then 121 degrees Celsius is very hot, because just to remind you, 100 degrees Celsius is boiling point of water. So the autoclave is a preferred method of sterilization for surgical, medical equipment, lab equipment. So when I used to do research in lab, I wouldn't grow bacteria in anything until I've sterilized it. So I used to put all the material that I was going to run an experiment with, so my flasks, everything, my agar, everything, I would stick it in an autoclave, and then once it was done, I would use it to make sure that I'm starting with sterile material. Another physical control method is filtration. So most of you know what a filter is, but it's basically, if we think of like paper, it's paper with little holes in it. So examples where we use filters are membrane filters used for water and air filters. And so this is a physical way to control microbes, because you don't let very, very small things in, depending on the size of the filter. So you could catch all the microbes on the filter so that your water goes through. Now, filtration doesn't really work for viruses, because viruses are very, very small, much smaller than bacteria in general, but They're very effective in filtering out a lot of bacteria and other microbes. Another physical control method is radiation. So what radiation actually does, whether we think of sunshine or which is UV radiation or UV radiation that's artificially made in labs, so you can buy UV light. What it does is it damages DNA, and by damaging DNA, you kill the microbe. The way it damages DNA is it creates these things called thymine dimers, meaning it gets thymine to bind to thymine, and this causes a break in the DNA, and the DNA is just not effective anymore. So that's why UV radiation is done. And I know a lot of people are using UV radiation right now for various ways of controlling coronaviruses. I saw online and on Amazon, people are buying UV lights. This is the same thing with sunshine, X-rays, gamma rays. So sunshine a lot will kill microbes because of creating thymine dimers by damaging DNA. So this is also why sunshine over a long time can cause skin cancer, because of all the mutations done to DNA, all the changes you do to DNA. Sunshine and acne, there's a lot of studies that have shown that people who have a lot of acne on their body or their face, it can be reduced by sunshine, because remember, acne a lot of times is caused by bacteria. So if you can damage the DNA of bacteria, you stop their growth. But also keep in mind that you're also damaging your own cells. So it's kind of, you have to weigh everything out. And then I always tell my students about the gel manicure trend. So with gel manicure, people are sticking their hands under radiation, and that radiation is also damaging your own cells because it's creating thymine dimers over a long period of time. So keep that in mind. Now, microwaves are not radiation. They kill bacteria because of the heat, but it's not, they're not high radiation. So just keep that in mind. They're not antimicrobial. So those were physical methods. So we talked about heat, which denatures proteins and other cell components, filter, which just physically filters out things, and then radiation. And what radiation does is it damages DNA, it creates thymine dimers. Now we're going to talk about chemicals that people use every day to control microbial growth. So chemicals can disrupt cell membranes by affecting lipids. That's what a lot of the different cleaning supplies that we do, that's how they hurt coronaviruses, because coronaviruses are enveloped viruses, meaning they have a phospholipid bilayer, and that phospholipid can easily be disrupted by various chemicals, such as alcohols. Chemicals can affect proteins, enzymes are proteins, they can affect nucleic acids. So here's an example of what chemicals do. Alcohol, so hand sanitizers are very high, most hand sanitizers, not the natural ones. Actually, the natural ones are not very effective if we're talking about controlling coronaviruses, because a lot of times the natural ones are oil-based and not really alcohol-based. What alcohol do is they denature proteins and they dissolve lipids. And so alcohols generally have no effect on endospores, and some viruses are not affected by alcohols, but we're very lucky that coronaviruses are enveloped viruses. We'll learn about it later, but some viruses are naked viruses, some are enveloped viruses. Enveloped viruses are easier to treat with chemicals such as alcohols because they have that lipid, and alcohols denature proteins and dissolve lipids. Chlorine is an oxidizing agent, so it causes different, it's very reactive. Oxidation means to lose electrons. So by being so oxidizing, it changes the structure of different cell components. Lysol breaks down cell membranes and denatures proteins. That's why we use Lysol to affect different microbes. Bleach, what bleach does is it causes cell components to be very reactive. So acids and bases, remember, one is a proton donor, one is a proton acceptor. They're so reactive that they ruin the structure of different cell components. And then there's many, many others. And with all the chemicals we use, there's a concern of overuse and misuse. So right now, with everything going on with coronaviruses, we are way overusing these chemicals, and a lot of studies are coming out, and I think we're going to see more in the future after hopefully this year passes and coronavirus hopefully will pass. What I predict and what I've been reading is that we're seeing so many resistant bacteria come up because of the overuse of chemicals. So we're trying to really control one microbe, which is coronaviruses, but overusing chemicals is also not a great thing, because then you get a lot of resistant microbes come up, and when resistant microbes come up, you have to have new chemicals that work that we currently don't have. So a lot of research has to go into antibiotics, which we will learn about later. Another way to control microbial growth is through preservatives. So preservatives are added to food to prevent spoilage. And so a lot of times we use sugars and salts as preservatives. We use sugars and salts as preservatives because they create a hypertonic environment. And remember, cells shrink in hypertonic environments. We also have organic acids as preservatives. The reason we use organic acids, if you've ever read sorbic acids, benzoic acids, many different acids that you can look at food labels. So next time, read your label. A lot of times we see these in yogurt, is that they inhibit microbial metabolism. And if microbes can't do their metabolism, they can't get energy, they can't grow. We also use nitrites and nitrates to prevent endospore germination. So if endospores go back and germinate, and germination means that you go back and you're able to be a reproductive bacteria, that's not good. So that's why a lot of nitrates and nitrites are used. Now, these can have their own issues, their own health issues in the long run, but in general, we're using them to prevent microbes from growing. Okay, so just to go through these, do pickled foods spoil fast? And the answer is no, because it's a hypertonic environment. So by hypertonic environment, you don't get a lot of microbes to grow. Same thing with salted food. I'm sorry, pickled foods are salty and acidic. So that was my salt reason, the hypertonic environment. But pickled foods also don't spoil fast because of very low pH and acidity. And remember, most microbes are neutrophils. Okay, and then another very important thing we use to control microbial growth is antibiotics. We're going to learn all about antibiotics later, but antibiotics are used in food and in human illnesses, and we use it in food production to not get microbes to grow. Antibiotics can affect many different things, but antibiotics, depending on the antibiotic, typically will affect the cell wall synthesis of bacteria, they'll affect protein synthesis, they'll alter the cell membrane, and they can affect RNA and DNA, which are nucleic acids. So when you take an antibiotic, you're not only affecting the bad bacteria that's causing an infection, but you're also affecting the good bacteria. And they can also affect metabolism. Okay, and then finally, I want to talk about hand washing versus hand sanitizing. So hand washing is to limit microbial contamination. And I want to tell everyone that hand washing is way more effective than hand sanitizing, because most people do not sanitize their hands properly. That's the reason. So hand sanitizing properly is putting a good amount of hand sanitizer and letting it sit on your hands for a while. Hand washing also, a lot of times people do not wash their hands effectively for the amount of seconds you're supposed to and the amount of soap and the temperature of water. But both are really great ways to control microbial growth. That's why we do these things. That's why right now I feel like a lot of people are more aware of hand washing and hand sanitizing with coronaviruses and COVID-19. So just like we're using it to control COVID-19, we can also use it to control a lot of different infections. So most infections in the world, we're going to learn later on, have gone down because people have learned how important hand washing and hand sanitizing is. I always ask my students in lab, how do we test which method is more effective? And it's not about which is more effective or which is less effective. It's more about if you're actually doing it properly. So if you wash your hands for 2 seconds, that's very different than washing them for 30 seconds. If you hand sanitize your hands really quickly with a tiny bit of hand sanitizer, that's very different than using the proper amount and letting it sit on your hands for a while. So the way we test which method is more effective is You can get one person to wash their hands and one person to do hand sanitizer, and they can do a before and after on an agar plate. So here are dirty hands on an agar plate. Basically, you just take an agar plate and put your hands. Sometimes we do this with students, and then the person washes their hands and they go back, and you can see that the dirty hands grew so much bacteria. All of these are colonies of bacteria. The clean hands still had bacteria, but it went down. And we could do this to compare also hand sanitizer. Now, we are seeing a big fear of microbial resistance with the overuse of antibiotics and various cleaning methods, acne medication, all of these things that people are using antibacterial soap. So most people should not be using antibacterial soap. Antibacterial soap should be used by, for example, research labs and hospitals, but at home, all of us should be using regular soap because we're scared that all of these chemicals we're using will lead to microbial resistance. The way you get microbial resistance is if you have a group of microbes and you keep using antibiotic soap and maybe one resistant bug survives, it can reproduce and create a lot more of that antibiotic resistant bug. And now the antibiotic soap won't be as effective. Now, a big question is, do hand sanitizers lead to resistance? And as of right now, we're saying probably not, because they're alcohol-based. And alcohols target so many different things in a cell. So it's going to take a lot of time for organisms to become resistant to all the different components that alcohols target. And so I wanted to end the slide with how do we control coronaviruses. So this novel coronavirus, so this slide, it was called novel coronavirus when I made this slide a few months ago, but now it's called SARS-CoV-2. So a virus is a microbe. We can control its growth, and what we're doing right now, as is the whole world, is washing hands frequently, hand sanitizing, cleaning really well, cooking food. Can you cook it off? Yes, cooking food is very effective for all microbes, not just coronaviruses, but just keep in mind that as we are seeing right now in the world, how we control various microbes, we do throughout our life for all the different microbes. So here are the study questions for you. So basically we started off this lecture talking about bacterial growth, about biofilms versus planktonic cells and what quorum sensing is. Then we talked about growth curves, and remember the 4 phases of bacterial growth curve is log phase, stationary phase, and death phase. And then we talked about what's important for microbes to grow, so temperature requirements, salt requirements, pH requirements, nutrient requirements. And then after learning all of these, we learned about how, once you know all of these, how do we control microbial growth? So we talked about physical methods and chemical methods. So physical methods such as heat, radiation, filtration, and chemical methods such as using Lysol and alcohol and various alcohol-based sanitizers. So go through everything. I know it's a long lecture, but just take time. And I hope that everyone leaves this class and this lecture knowing what the word sterilization means. So if something is sterile, it means it has absolutely no microbial life on it.